working. A technician, please help me. Okay. I'll just use my voice then. Oh, thank you. It feels like stand-up comedy when I have a microphone, and I really like that. So, thank you. You're a biotechnology people, right? Right, you are. Oh, that's really unfortunate because I thought you were physics people. And I had prepared a joke that worked really well if you all had been studying physics, but you haven't. So yeah, you like physics? Okay, I'll try it then. Okay, so I didn't really have a joke. So okay, we'll just play a song then.
Please join me in giving Strindens Promenader-Kassett another round of applause. Well, I just wanted to say I was really excited to hear that physics joke. I don't know exactly what that was going to be, but I can give you a quick biology joke. Why did the biologist break up with the physicist? They had no chemistry. Okay. Anyway, welcome to this year's Digital Life Conference. It's the 7th year that we're having this conference, and we're back in Trondheim, where the first conference was. Before we get started, a couple instructions for if there's an emergency situation. The gathering point is out in the parking lot outside the hotel. There's 4 emergency exits in this room, and all the emergency exits are marked with green signs. So again, thank you for being part of this year's theme, which is how to work transdisciplinary in biotechnology. If you know Digital Life, and I guess all of you do, you know that transdisciplinarity is a central part of what we work with. It means working across disciplines and working across sectors outside of academia and inside, across biology, biotechnology, chemistry, physics, computational biology, and a lot of different mixtures of those and other fields. So one of the missions of Digital Life is to stimulate transdisciplinarity in education, research, and the work that we do across the country. We want to help create pathways for ideas to come out of academic projects to stimulate innovation, to create value in a sustainable and a responsible way, and to create pathways for young researchers to continue their careers in biotechnology in many forms. So we have put together what I hope is a great program for you with a lot of different perspectives on transdisciplinarity and biotechnology this year. And I'm very excited to start off with Lisa Bitz, who will be joining us in just a moment. She will be delivering the 2022 Volterra lecture, which Digital Life Norway is resuming this year after a bit of a pause in a different format. We are bringing it back here at the conference. Vito Volterra is the person that the lecture series is named in honor of. He was one of the first mathematical biologists who became a professor of rational mathematics at the University of Pisa in the late 1800s, where he contributed to developing a theory of functionals and integral differential equations. The Volterra lecture series from Digital Life Norway has been hosted since 2016, and we're pleased to use this opportunity to highlight the evolving nature of digital biotechnology, including its responsible research and innovation aspects. So about today's Volterra keynote, Lisa Bitz is a senior project manager at the Danish Board of Technology, where she's the work package leader on responsible research and innovation for the EU flagship project, the Human Brain Project, which is a Europe-wide effort to pioneer digital brain research to help understand the complex structure and function of the human brain with an interdisciplinary approach at the intersection of neuroscience and technology. Lisa has also been coordinator of the EU project Go Nano, and so she brings experience as well in nanotechnology, and she specializes in citizen and public engagement in science and technology. She has a PhD in science and technology studies from Twente University in the Netherlands. Please join me in welcoming Lisa to the stage.
Hi. So thank you first and foremost, Cam, and thank you to the Center for Digital Life Norway for having me. It's really an honor to come here and to talk at your conference. I'm really excited today to share the work that I've been doing and we've been doing within the Human Brain Project on public engagement. It's also a little scary because as an audience, you're different than the audience that I'm used to, so I hope that you will find some interest in my take on transdisciplinary work. So what I'll do today is start with a little background about the Danish Board of Technology Foundation and about public engagement, what it is, how to do it and why to do it. Then I will show an example of work that we did in the Human Brain Project around AI and data. And then I will end with a nod to some of the challenges that one will experience when wanting to do public engagement work, but I won't be completely negative because there are also some opportunities that I'll point to. And then, of course, I will tell you a summary, what were the key points, and invite you to the workshop that I'm hosting tomorrow on public engagement. So, in terms of background, now Kam already said most things about my background, but maybe just to add that I actually have a bachelor also in biotechnology from Aalborg University, so I'm sort of a crossover. So it's kind of nice to be here also, actually, in that sense. But the Danish Board of Technology, where I work, is a foundation that has as its stated mission really to be transdisciplinary, to work for a society where its development is shaped by what we call informed and forward-looking collaboration between citizens, experts, stakeholders, decision makers, and citizens, both internationally and nationally. And we do that work primarily through what is called public engagement. And we do that on complex issues such as science and technology development, health, welfare, and climate. And in that process, our aim is also to contribute to developing more democratic formats for decision-making processes. So we work really in this cross-field between science and policy. Now, what is public engagement? One way of looking at it is to think about who is it actually that we are wanting to engage. Who's meant to be engaged? And here we can separate, and we're not the only ones who make this separation. It's also separation that the European Commission works with between experts and stakeholders and lay publics. And these audiences, they sort of belong to sort of four spheres of societal engagement, one could say. So there's research, policy, civil society organizations, NGOs, so interest organizations, and also, of course, industry and business, and then the lay public. And public engagement is really about using these sort of overlaps between these spheres to create productive spaces for learning. And so one can organize, for example, expert or multidisciplinary engagement between different disciplines, so involving policymakers or other kinds of experts from civil society. But when you add sort of lay public to the mix, then it's citizen engagement and becomes what in my understanding would be real transdisciplinary work. And the key point of doing this is really that many of the issues that we face as a society are complex, and therefore representatives that move in these different spheres of society, they have different knowledge, they have different experiences, different takes on what an issue or problem is actually consistent of, and can therefore also contribute towards understanding what a good solution would be. And then finally, and maybe one of the key points to get across is that when we talk about public engagement, it's about giving influence. So all of the actors that are involved in a public engagement process should have an opportunity to also influence the process going forward. And that also goes for involving lay citizens. So when you do citizen engagement, involve them in a process, they should also have the opportunity to influence priorities and choices in the process that you're involving them in. A little bit more about why. So there are three sort of main arguments coming from a theoretical point. One is that science is a public and to some extent also a political activity, and therefore it should be more open to democratic control, and we can create that by opening up decision-making and prioritization making processes in our societies, and we are then aiming to develop ways of doing that. Public engagement is one of them. And as I just said, citizens and other stakeholders, like coming from policy, industry, research, civil society, they have different experiences and different kinds of knowledge, valuable knowledge that can really help understand issues and find new ideas and solutions. And also involving a broader and a more diverse audience can help create a better foundation for decision making, either in a science development process or in policy. Then practically, to take it down maybe a little bit from the theoretical, really the why comes from this possibility to really better understand what a problem is, but also to get insight into potential conflicts or ongoing conflicts. What do they actually consist of? How does the conflict look from different perspectives? What are the values at stake, needs, concerns, and so on? So really an engagement process can also help you understand that. And you can also get new ideas then through this process, because you might see things differently than you did before if you engage in a dialogue with audiences that you might not always or necessarily before engage with. So you develop new solutions, and you can also learn if you are wanting to take a decision, and for example, if you're in an implementation phase with something, do I go in this direction or do I go in this direction with developing a device or new treatment or a new approach? Then you can learn what direction has the most support. And finally, engagement processes in themselves are also really useful for building communities and networks, and for getting to know new people. When then to engage? Well, maybe the good news is that you can do it whenever, wherever you are in your process. Be it either sort of policy or vision formulation in terms of what it is that you would like to do, program development. Are you defining the projects? Are you actually engaged in a research activity? Are you trying to implement something coming out from research? The but is that there's sort of an inherent sort of contradiction or difficulty in this process, is that in the beginning, when you are formulating visions, the possibility to influence directions is really high, because nothing is really sort of set yet and nothing has been settled. So there's a high degree of possibility to influence. But when you, the contradiction is that the motivation to engage then in the early phase is lower, because it's harder to see what will actually be sort of the implementation, what will actually be sort of the effects of what you're trying to do. For example, if you're trying to engage lay publics on their everyday life, if they can't see like an immediate effect on their lives, it's really hard to create interest and to get them engaged. So the motivation to engage increases when you get closer to an actual use context. This next slide is just to give you a quick overview of some of the many different ways in which you can do engagement processes at many different levels and at the different stages of a process. What matters really the most is that you choose the right approach for what it is that you would like to achieve, get new ideas, insight into a conflict, or whatever it is that suits your need. And tomorrow I will show you sort of a catalog of methods that can also be more useful in terms of trying to pick the right approach. Now, to the example from the Human Brain Project. I don't know how many here know the Human Brain Project, but as Cam just said, it's a big flagship project funded by the European Commission. It is one that's been running for almost 10 years. So it started in October 2013, and we are ending in September next year. So we got a little bit of an extension because of the COVID-19 virus, but we will be ending pretty soon in the final stretches of that project. And that project is one that has changed a lot, I think, over the 10 years. And now, in the final phase, eBrains, as we call this European research infrastructure, a digital research infrastructure, is going to be sort of the main legacy of the project. It's an infrastructure that is created to promote digitally supported brain science, development of medicine, and computing technologies. And in this final phase, our tasks in the Human Brain Project is, as Cam said, to lead this work package on responsible research and innovation. And what we're really focusing on there is to ensure the legacy of all the work that we have been doing in the RRI work package, but also in the Ethics and Society project that came before that work package, and seeing how we can ensure the legacy of that work and practices, principles into eBrains as a research infrastructure, because a research infrastructure is something quite different than a research project. And then we are also collaborating with neuroethics colleagues, and also through them other brain initiatives around the world, to look at neuroethical issues and see how we can develop an international strategy for how to deal with present day and emerging neuroethical issues also in the future. Then we develop citizen and stakeholder engagement, which is the example that I'm going to give you today. And finally, we also have a task in terms of building a community for eBrains, because the eBrains research infrastructure is based on principles of co-creation. So the idea is really that the platform services will be co-developed with the user community, and we have a task there in terms of helping to build this community as an inclusive community. Now, the example that I'm taking with me today is a little bit old, in the sense that it is work that was started in 2018. And just, I don't know if how many can remember the conversation around AI and data use in 2018, but it was quite different than it is today. Really, a lot of things happened in that debate over the last principles, responsible use and development of AI. And we wanted to create make a start in terms of thinking about how can we actually handle, be more proactive around these kinds of issues within the project, also looking towards eBrains. And finally, we wanted to work on strengthening capabilities within the project to work multidisciplinary and transdisciplinary. So the methodological approach that we chose was to start with a multidisciplinary workshop of experts, asking experts with various backgrounds to go through multiple criteria and dimensions and ask them to assess possible future impacts and come up with possible solutions. And the next step then in that process was to use the input from the experts to go through a citizen engagement process and ask citizens about their reactions to the expert input, what their needs and wishes and desires would be in terms of future developments. And the third step became a little bit delayed because of COVID-19, so we plan to follow up with national and international policy workshops or expert workshops. And those workshops are happening, one of them the end of this month and another one in spring. So this first step, AI 360 Copenhagen, was held in 2019. And maybe can be hard to remember, but as I just said, the conversation around AI was very different at that point in time. And the main debate was really around what's going to happen to the future labor market, what kind of skills are needed, how can we make sure that we don't have a big portion of people that fall off the employment market. And we wanted to create a better overview of what are the actual future potential issues that we need to be anticipating on. And so I'm showing you here some of the results to give you sort of a feeling of the breadth of an overview that can be created within one single workshop where you engage multiple dimensions and assess possible future impacts. And one of them was policy, and some of the things that they pointed to there were concerns around manipulation, abuse of power, about, for example, this question, who decides what is good information and correct? And they also pointed to concerns in terms of power relations between state and private actors, issues that are also relevant today. And these were actually some of the concerns that they couldn't come up with really good solutions for. Then we looked at the rights and law sphere, and some of the issues that they were really concerned about here were, well, enhancement of ongoing discrimination issues, but also new forms of discrimination, bias, and the impossibility of holding, for example, social media platforms accountable for the effects that they have on societal dynamics, democratic institutions. And also they pointed to the issue of surveillance. It's really hard to get an overview of who's surveilling who and why. They pointed to a huge lack in terms of education on data governance and data protection, also at the, let's say, in the individual level, how to protect oneself, but also, organizations, how to protect themselves. And then the impossibility of getting insight into what data private actors actually collect. Then they also went through economy and more general society. And in terms of economy, they pointed to one positive feature, that we would have a lot more free services somehow in the future. The question, of course, would be who will enjoy the benefits of offering those free services? Who is it that's going to make the money? Who is it that's going to become powerful? What is it that data is going to be used for? How is that going to be distributed in society? They pointed also to like a more intense data economy. What about maintenance and the cost of implementation of all these new systems? And they also pointed to this contradiction between how to stimulate innovation and at the same time respect individual rights. They thought that was not a given, that that's going to happen, both of them together. And finally, they pointed also to a challenge with sustainability and productivity, that doesn't necessarily go hand in hand. So you can see there is really some climate questions also. In terms of society, they pointed to the great potential for diagnostics and personal medicines using more sort of AI-based systems. But the question that they pointed to there is really how do we ensure that use is correct and safe? How do we ensure good data quality and also maintenance of databases? They pointed to that one must not forget that even though you have smart systems, there's still a need for someone to be able to have a good understanding of disease mechanisms and be able to explain those. And then they also warned against going for a specific solution before really understanding what the problem is that one is trying to solve with for example, an AI-based solution. Now, then to the public engagement activities. So this multi-stakeholder workshop was then followed by what we call dialogue meetings with citizens in 13 different European countries. The citizens met in their own homes or other sort of comfortable settings in groups of 5 to 8 people and carried out dialogue supported by an online platform that's called Engage Suite. And there they're giving sort of short text to read about some of the issues, videos, introductions. They're given some time to discuss with each other about the issues, and then finally some questions that they answer. And we had in total 157 meetings across these 13 countries, and were able to engage 928 lay citizens in dialogue. And our main findings on these meetings, just to show what their sort of joint recommendations were, they thought AI should be a decision support tool, should be regulated, certified, should be explainable, understandable to them, but also to the experts who use the AI-based solutions. And then they shared this concern on political use and social manipulation, and some of them even shared how they already, adapt their behavior online because they're worried about who's collecting their data, who's using insights about them, and for what. So just to wrap up the main outcomes of this exercise, we found together both from the experts and from the citizens this concern about misinformation, social manipulation and effect on democratic institutions, desire for regulation and control, and demand for human intervention and explainability. And also these exercises really were able to show that development and implementation of AI is about much more than future discussions about future labor market. There's much more at stake here in terms of how we govern and steer this development. And this is a point to really take back to a project such as the Human Brain Project, a big project. How can we also help to take some proactive initiative to steer these developments? And then was the case then, is probably also the case now. There's both a lack of information about the potential of developing AI, but also a lack of information about all these issues around the use of data and data governance and data protection and collection of data. So what did we achieve within the Human Brain Project? As we set out, we wanted this overview, so that's what we got, and also perspectives and possible solutions that, of course, go beyond the project. In the project, we got a better understanding of some of the legal challenges, how we could exercise good data governance, ethical issues, and related societal questions on value and benefit. Concretely, we have developed what we call eBrain's vision on ethics and society, and that is something that we are trying to take the next steps in terms of figuring out how to implement in the research infrastructure. But this vision is really based on all of the work that we've done on responsible research and innovation within the group. Then we're working on setting up a committee, so a group of experts that can advise eBrains on ethics and societal issues going forward, also to continue to be proactive. And we have developed a number of capacity and training modules, of course on public engagement, but also more broadly on ROI, animal research issues, neuroethics, and the whole breadth, let's say, of work that we have taking place within our research group. Now, I said I would mention some of the challenges, and here they come, some of the depressing challenges, maybe. One of them is around the rationale, I think, for public engagement. That's definitely something that I've come up against a lot over the years. Why should we do this? Why couldn't we just sort of continue in the way that we've always done things? And so that's really something, I think, where we need to have more conversations in terms of reaching a more joint understanding of the why. Then also there's a lack of knowledge. I mean, public engagement is not an activity that is really widely known, I think. So that's also maybe a problem. And that is related to the fact that we have often, let's say, a tradition of working in more sort of siloed communities. So we simply do not know each other, and many scientists are also brought up in very sort of specific ways and educated to think in very specific paradigms. Then there's some very important point on funding. So it can be really hard to find funding to do transdisciplinary work or wanting to engage public or do something a little bit different than what everyone else is doing. Time. I showed you a process that we started thinking about in 2018, and where we had, okay, because of COVID also, but where we're taking some of the final steps in that process this year and next year. So it takes a lot of time and preparation. And well, there's also, I think for many young researchers at least, a question on career impact, because is this an activity that if I spend time on it, is that something that will be valued, that I can use for my future? So those are some of the challenges and concerns for doing more public engagement. Some good news, I think this conference is actually an example, and the Center for Digital Life is an example of some good news, because I think these conversations between communities and also on engagement and how to do sort of more multidisciplinary work is really being stimulated here, but also at the European Commission. The new framework program, Horizon Europe, really emphasizes multidisciplinary work, collaborations with social sciences, humanities, and also public engagement work. So I come from a foundation that's not a university-based, but we call ourselves practitioners, and there are many like us out there who are really very experienced with organizing and doing these things in practice. So these collaborations are really encouraged by the European Commission, and there's opportunity to find funding there. Also, I think it's just to show that I think it's really a priority from the Commission. There's recently been this big conference on the future of Europe, and we are right now also involved in organizing some follow-up to that for DG Research. that wants to involve European lay citizens also in coming up with priorities for the next round of the framework program. So this is really an avenue, I think, that is opening up more and more. So just to begin to wrap up, the key points from today is something, yes, that I come up against a lot. Can citizens engage on complex science and technology issues? They can. Of course, you have to explain in the right way, but they can understand and debate complex issues, and they actually really want to be engaged. Obviously, I think multidisciplinary engagement can bring a lot of value, as also this quick overview that we were able to create shows. Then public engagement, it can be used for many purposes, from vision to implementation, and there are really a lot of different opportunities for doing it and designing processes. Thank you for listening, and as I said, tomorrow there's a workshop on engagement where you can learn more about public engagement, try out some techniques and practice, and maybe even start to design your own proposal or exercise. Thank you for today.
Thank you so much to Lisa for that presentation. We're going to take a short technical break to prepare for the next session, so please stay in your seats and talk to the person next to you, and we'll be right back. Alrighty. Short technical break. It was a short chat break. I hope you enjoyed the opportunity to get to know some other people who are here at the conference this year. So on behalf of Digital Life, we are really pleased to have a session this year featuring EMBL, the European Molecular Biology Laboratory. And to introduce the session, I will call up Line Marianne Grønningvang, who is a Norwegian delegate to the EMBL Council and based at the Research Council of Norway, where she has responsibility for coordinating the Council's investments in life sciences. She also received a PhD in medical biochemistry from the University of Oslo. Line.
Hello, everybody. So yes, welcome to this session on EMBL. And this is really the European flagship for European molecular and cellular biology research, infrastructure, method development, scientific training, and knowledge dissemination. And EMBL was formed in 1974. And EMBL is world-leading within a number of fields, including bioinformatics, structure biology, genomics, and molecular imaging. EMBL is an intergovernmental organization with 27 member states, and Norway has been a member since 1985. EMBL recently published its new and ambitious 5-year program, From Molecules to Ecosystems, with the aim of establishing the molecular basis of life in context, to gain new knowledge that is relevant to understand life on Earth, and to provide translational potential to support advances in human and planetary health. The new program aligns very well with several of Digital Life's key priorities, such as transdisciplinary research collaboration and approaches enabling fair sharing of data, data analysis, and modeling. Digital Life works closely with Elixir Norway, the Norwegian node of Elixir, the pan-European research infrastructure for bioinformatics, an ESFRI landmark. Elixir works closely with EMBL EBI, the European Bioinformatics Institute, on training, standardization, and access to specialized data resources. So we are honored to have with us today Ewan Birney, Deputy Director General of EMBL, and Joint Director of EMBL EBI. I'm sure most of you here today are using EBI's freely available data resources and tools such as Ensemble, UniProt, BLAST, AlphaFold, to mention just a few. Ewan has been central in Genomics England and is a strong advocate for making genome information freely available to all. And Ewan will give a presentation on the impact of EMBL and EMBL's new program for European life sciences. And after Ewan's presentation, we will have a panel discussion on how EMBL can meet the needs of the life science communities in Norway and what role Digital Life can play towards EMBL. And joining Ewan then in the panel discussion will be 3 Norwegian alumni that I will introduce later on, and Scientific Director of Digital Life, Trygve Brautaset. But now, please, Ewan, the floor is yours.
Great. So thank you so much for that introduction. I'm having a tour of Norway. Yesterday I was in Oslo speaking to the medical community and the research community there. Today I'm in lovely Trondheim. My family actually has paid respects to King Olaf's Cathedral and St. Olaf. And then tomorrow we go to Tromsø, so that please pray to Norwegian weather gods that we can see the Northern Lights, I hope, but if not, we'll have a great time up there. And then I'm speaking actually on my research in Tromsø University on Monday. So if you want to hear about my research, fly to Tromsø, which is quite an ask, or persuade them to live stream it, which might be easier. But I'm going to speak now really about EMBL and EMBL's history, EMBL's current aspect, and above all, EMBL's relationship to Norway. I am 1/27th legally Norwegian, so I have my discovering my inner Norwegian at the moment. And you can think of EMBL as there being 6 little Norwegian flags planted in Cambridge, UK, in Heidelberg, Germany, in Hamburg, Grenoble, Barcelona, and Rome. And you're very welcome in all of those locations. So this is just a little bit of history of both molecular biology and EMBL's role in this. EMBL is a sort of child of the 60s. That was the time where 2 things were going on in science. So the first was the development of these powerful new physics machines, these machines which develop new particles and new systems, and most obviously led to the development of CERN. And there was an understanding, in particular in Europe, that one could only do that by collaboration between countries. Just the scale was going to be unfeasible. And the second thing then was the kind of development of both structural and then molecular biology at the time, and an understanding that these new types of structured light, very, very synchronous and bright X-rays, very powerful protons and neutrons would give us new insights into the way life works. And that has certainly been true, in particular for X-rays. And the second thing is that there was a new science at the time, molecular biology, and there was tremendous concern that that science was going to be completely dominated by America. And so they wanted to make sure that there was a flagship European laboratory to do that. And because of that, EMBL was founded in 1974 in Heidelberg, Germany. It was founded next door to a neutron source. Now, neutrons are lovely, especially if you're studying magnets. They're kind of useful for life, but they're not tremendously useful. And in fact, the neutron source closed down in Heidelberg. So left up on the hill is this massive molecular biology laboratory there in Heidelberg. But right at its foundation, it was joined by sites. There's a question about whether you brought the biology to the instrument or the instrument to the biology. And for these very big physics instruments, it was very clear that you had to bring the biology to the instrument. And so it was started with 2 outstations, or now sites, at Grenoble and Hamburg, which were the sites of still the two best synchrotrons in the world. Grenoble is the best synchrotron in the world and produces the most synchronous and powerful X-ray light for people to use. And if you go to Grenoble and do your crystal structures, you'll be using one of the beam lines run by EMBL, or if you go to Hamburg. And then we go through the 1980s and really the discovery of all the different things you can create via molecular biology, and understanding life now by thinking about its DNA and mutations to the DNA and how it comes about. And most impressively was Christine Nusslen-Volhard and Eric Wirtschas were at EMBL when they did the Drosophila screens, which won them the Nobel Prize later on. That's not the only Nobel Prize that is associated with EMBL. Indeed, at the same time, Jacques Dubochet developed cryofreezing, so the ability to flash freeze biological samples and to do it so quickly that the water does not crystallize. So the water is in this glass state. The water molecules are trapped in that instant, and therefore you do not disrupt the biological nature of the material you have. And that flash freezing was absolutely critical for the development of cryo-EM, which has become such a mainstay of molecular biology, structural biology. Underneath all of this was a theme of DNA and informatics. And this grew and grew over the 80s and the 90s. And at the end of the 80s, the scientists in Europe, particularly in Heidelberg, Ryan was there and some other people were knocking around, were taking journal books and typing in the DNA sequences that were being published at the time. So they're typing them in directly. A human being was reading a paper typing it in, and then that would go into a computer. This was before the internet. And so what would happen would be that if you wanted that DNA sequence, you would order it, and you could order it in two forms at the start. One was printed books. You could get a complete printout. And so I have seen the shelves of this. It's kind of mad, but brilliant at the time, a real sort of 1980s solution to the problem. Or you could order tape drives. And just when I was just a kid, I was knocking around in this era, and it was very exciting about CD-ROMs. We could have CD-ROMs, and we could send CD-ROMs in the post. So all of this business of sharing data with our colleagues in America and in Japan, it predates the internet. It's quite important to stress that biology's attitude towards data sharing predates the way society has learned to use or exploit data sharing and communications at scale. Now, back in the 1980s, this got out of hand, and the people in EMBL put their hand up and said, we need more space for these people typing this in. It's all getting a bit more serious. And EMBL Council at the time said, well, there's no reason why that should happen in Heidelberg. And so countries were invited to bid for the next site of EMBL. And 3 bids went in. One went in by Germany to expand the site in Heidelberg. One went in from Sweden, which was going to be based in Uppsala, which would have been rather lovely. And the third one was in the UK to place Europe's bioinformatics center next door to Europe's largest sequencing center, the Sanger Center. And that was, the campus was funded by the Wellcome Trust, so the building was a gift from the Wellcome Trust. So the UK did win that, and that's why EBI was present there. And just to finish off the whole story of how the sites came about, at that point it became clearer that it was feasible to run a multi-site molecular biology institution, and we started a site in Rome around mouse biology. It's now focused on neurobiology and epigenetics. And then our latest site is in Barcelona on the beach. Absolutely beautiful building, focusing on mesoscale imaging. And you can see now, as we go through to the 2000s, 2015, 2017, and onwards, we have continued at EMBL to be part of innovation. Sometimes that innovation is innovation that happened 20 years ago that we've been recognized for now, and sometimes it has been this incredibly rapid and fast innovation. For example, the creation of the AlphaFold database across all known proteins, applying this AI method to produce accurate structural prediction models for every single protein that we know about. And we believe with our colleagues in DeepMind that we'll be able to stay up to date for that for the foreseeable future. So that's a bit of a history. We have underpinning that history are these 5 different missions. One is excellent research, to perform excellent research and to be that flagship in Europe for molecular biology. The second is to provide scientific services. Now, these are most obviously the synchrotrons, the beamlines for X-ray crystallography. If you do X-ray crystallography, I'm sure you know about these beamlines. They include now the EBI data services, and that has become a linchpin to molecular biology across the world, including Europe, about these data resources that we show to the world. And finally, the newest addition, which I'll touch on later on this talk, is the Imaging Center in Heidelberg. And that is access to super resolution microscopes and to EM technology for, again, all member states. You can put your hand up and go in a queue to access those machines. And then we have 3 supporting missions. One is advanced training. That means students and postdocs. We have a very vibrant student and postdoc rule. And back in 1974, the countries that founded us said that you must not keep people. And so we are not allowed to hire from our student population any postdocs. And we're not allowed to hire from our postdoc population any faculty members. And our faculty members must leave after 9 years. There's a small set of people who are considered tremendously important or lucky, who go beyond those 9 years. But legally, over 90% of people must leave beyond 9 years. And this means that EMBL is always turning over and always producing people. It also means for whatever career stage you're in here in Norway, there is a career path that involves EMBL. So if you're interested, there will be a studentship or a postdoc or faculty position. And I'm really, really keen for you guys to think about your career path as weaving into EMBL and then coming back, probably to Norway because it's lovely, but coming back somewhere else for this. Innovation and translation, again, I'll touch on it, which is about how we interact with companies. And then finally, integrating life sciences, and that's particularly true about our role with Elixir in data. And this is a picture of our lovely member states. We're a bit like Eurovision in the sense that Australia wants to be European here, so Australia is down at the bottom. It's the vast majority of Europe. Slovenia is a bit of a holdout, and we will get there at some point, I hope, joining together for this. And this slide is a busy slide because it also indicates all the partnerships we have. And these are partnerships which are local to the sites that we have, or partnerships which are remote and have set up under the EMBL model. And those include the Norwegian Centre for Molecular Medicine, NCMM, in Oslo, which has been a tremendous success there. And that's part of the Danish-Nordic partnership, but also includes places like Brain City in Poland. And this, again, is just to remind you of these 6 different sites. So our headquarters are in Heidelberg, Germany. It's also our largest experimental lab. The second biggest site is EMBL EBI, just south of Cambridge, on the same site as the Sanger Institute, and we're completely dry there. And then we have Hamburg and Grenoble, which are the 2 synchrotrons, Rome, which does effectively mouse biology, epigenetics, and neurobiology, and Barcelona, which is about tissue biology. So we have many, many different links with Norway in many different ways. This is a 3 that we're particularly proud of. So a publication and analysis of microbial sequencing done jointly with NTNU and colleagues in EMBL. This rather wonderful business of, and I think this is with the SARS Center, this business of looking at the diversity of metazoan life in marine animals. And this is looking at sponges and the different types of cells in sponges. And these rather small marine organisms are kind of brilliant because you can put the whole organism into an EM device and look at them there. And on the right-hand side, the development of the Federated European Genome Phenome Archive. So this is about the responsible sharing of clinical data between collaborations. And Norway has been one of the founding nodes of this federation for delivering this. And as you saw on the map, we have 2 different partnerships here. One is very much on the medical side, the NCMM in the University of Oslo, and the second one is the SARS International Center for Molecular Marine Biology to do this. And again, the partnerships differ, but one of the things that the partners, I know many partners, appreciate is the ability to have and model their career paths across EMBL of this turnover. Not every academic institution can have a turnover model, but some academic institutions having a turnover model ensures that we have throughput of people and new ideas entering the field. The important thing, and what I think and there's worth doing. To do that, for biologists, for the, there's a whole wide field of nanometer biological material. Look at how to get, but you can do things that you must be really. And then and service there. So again, if you have a project where you can imagine that you want to track something in a tissue or in cells, you can do that in cell culture. You need to be able to get a fluorophore label into your biological object of interest. And that fluorophore label has to be super resolution compatible or MinFlux compatible. Then this is an option, and it's going to be remarkable what we discover with these technologies. And there at Heidelberg, they're also bringing together both microscopy and EM tomography. And so this is the ability to flash freeze a particular tissue of interest, and then to mill, using an ion beam, a particular slice of it, which has a biological event of interest. So a virus fusing to a plate, or a particular cell type that you're interested in. Now, to capture and know where that event is, you'll need fluorescence in a wide field to spot that event. You can then mill down to that particular place, and then you can take that tiny piece of glassy ice with your event of interest and put that into a EM machine and image that in the EM machine. Now again, that machine is 1/27th Norwegian at some level, and every Norwegian is as welcome as every German, as every Brit, as every Australian to it. You just have to put your hand up to gain access. Now, you may well be in the situation where you're like, oh, I just don't know, is it going to work? What do I do? I've got this interesting problem, but I don't know whether it's going to work. And here, the friendly elves of EMBL will be happy to answer your questions, and you can write them and say, I've got this idea, I can definitely get this label in, I can definitely see this, what do I do next, or which technology should I use next for this? So please, please exploit us. Please exploit us to the max. Yeah. The second place, which I'm sure all of you use us every single day, for this is our data resources at EBI. So Ensemble for genomes, Uniport for proteins, the PB for protein structures, Reactome for protein pathways, the literature, mass spectrometry in PRIDE, metabolites for metabolomics, and I've forgotten, we've got 43. There's a lot of them there. I'm going to talk a moment later on in particular about AlphaFold and give you the story of AlphaFold. It's a great story. I hope many of you use it. I'll talk, I hope you all use these resources. You'll be a bit nuts if you don't for this. And one of the things we can do is run a training course as well here with our colleagues here in Elixir Norway to make sure everybody can exploit this. This is kind of data has become the lifeblood of molecular biology. To make this work, it needs to be international, and we are the European side of those international collaborations that make all of this work. Our new leader, Edith Hurd, my boss, came to EMBL from Institut Curie in Paris, and she challenged us to do even more, which was a bit daunting. I have to admit, the first time I heard what Edith wanted to do, I was like, are you sure?
But it has been really invigorating. And that is to keep one foot wedded to this atomic, molecular, cellular world, but to work in collaboration with all these people outside of the organism, so the organism in context. So this is to think about how organisms interact with other organisms and with the environment. And I have a particular passion for one organism. Those are humans, and they're quite unique in the way they interact and create environments, but all organisms count here. And that, by the generous awarding of money from EMBL Council, means that we can realize that. It is not a small ambition, molecules to ecosystems. Just be reassured, we do not expect to do all of this within the walls of EMBL, by definition. Rather, we want to be, again, a flagship and a place of collaboration across Europe to deliver this. And there's lots and lots of interesting environments in Norway, both for humans and for all these other wonderful things, which I'm sure I'll meet up in Tromsø. So this is this new program. It is bold. It meets a lot of societal challenges, both health challenges, but also global planetary health challenges, about how we think about the planet. And again, collaboration and interdisciplinarity has always been a big part of EMBL. We've always had a lot of physics and maths alongside the molecular biology that we do, and that will only get deeper and deeper over the coming years. So these are some of the things that we expect to impact over time. Like many scientists, I celebrate the times we create impact in the world and we do something that changes things. I get a huge buzz out of making people healthier. I think it's amazing. But that's not actually how I organize my science. I organize my science by curiosity and what drives it. And these are the things which we believe we're going to make an impact on, but it's not a given. And I suspect our biggest impact is something that we cannot write down now. And that's the way this goes. So looking at ecosystems, but in particular the carbon cycles and weird and wonderful new carbon fixation routes, which is present in life, and that's very clear in the microbial systems that we've been looking at, is there's quite a lot of diversity in the fundamental elemental cycles of life. Thinking about biodiversity and thinking about how we measure it, how we capture it, and how we use it in our understanding of life in evolution. Thinking about the impact on humans and disease, in particular the interaction of microbes, and thus we create an environment for a large number of microbes in our skin and our gut, so we are an environment as well as being people. And then one particular headache that we've got is like a slow burn pandemic, is antimicrobial resistance. I don't know if anybody knows this, it's very depressing. We are running out of effective antibiotics, and if we don't fundamentally change our ability to find new antibiotics or manage the way bacteria are getting around our current antibiotics, we could go back to the 1910s, 1920s, where we did not have effective antimicrobials. And that would be a very bad world to go back to. So that's quite an important thing for society to get right, because it's a darker place without that. And just like here, we are going to do transdisciplinary research. Now, so we set this up with these core of the cellular biology, structural biology, developmental biology, and computational biology that we've always had, but we've added some other areas here to stimulate these things. I'm going to step you through the newer areas, but I just want to emphasize that the central part is still just as big and vibrant as ever for this. And just to mention that this 9-year turnover of groups means that the actual individual research that's happening at EMBL is always changing and refreshing. So if you knew EMBL in the 1980s, at some level it's still the same physical building and there's aspects of the same culture there, but there are very, very few people who were around then, now, or in the 1990s or the 2000s. So stepping through this, we have the planetary biology transversal theme to look at the planet. This has in some sense been piloted 5 years ago by this wonderful ship, research ship called Tara, manned by gloriously mad Frenchmen, sailing the oceans and sucking seawater through filters and capturing microbes and viruses and plankton on their filters. There are way more viruses than anybody ever thought there were going to be in the ocean. Like, way more is one sort of surprising take-home from that. But the bacterial diversity is mind-blowing in the ocean. They want to do that again, but they want to do this around the coastline of Europe, and they want to do this with a Embl truck, the Trek mission, going alongside the coast, beside the boat, so that we will be sampling the ocean or the beach and the bit of sea close to the ocean, close to the beach and the environment, all at the same time. So they've done this. They've done a pilot in Naples. It's definitely doable. There's quite a lot of interdisciplinary work here between ecologists and molecular biologists. Interestingly enough, they think the genomics is very routine and boring. I find the genomics quite exciting. But they're going to take cryo-freezing equipment on the truck, so they aim to freeze a lot of these animals as well in a format that they can put into the electron microscopes later on. So freeze these weird and wonderful living organisms as well. And there's many other projects here, including our collaborations with the Earth Biogenome Project, which links up to sequencing all the animals on the planet. Again, one of those rather ambitious statements that you think to yourself, is that doable? So one, not every single individual, obviously, but one of every single species on the planet, every single animal, is a feasible thing to do. And a number of projects have started to do that in a number of locations. I would encourage, I would say Norway, but it's really the Nordics, to think about that as something to do across the Scandinavian peninsula. As I said, one of my favorite animals is humans. They're great animals, bipedal organisms. Humans are kind of interesting for a number of reasons. So one of them is they're so important for medicine. So understanding molecular biology of humans and understanding the physiology of humans is an absolute sine qua non for medicine. They also have this incredible ability to walk into phenotyping centers, which are called hospitals, and you can phenotype these organisms almost for free if you're a researcher by tickling the hospital just right there. It is a remarkable property of humans, and it means that some fundamental pieces of research we do basically around humans. My favorite humans are wild Danes in their Danish environment, or wild Brits in their British environment, because those are the two best tracked sets of humans around. But humans interact with an environment, and there's two types of environment for humans. So one is what you normally think about the environment, which is sort of temperature and chemicals and things like that, and that's quite an important environment and has an impact on everyone's life. The other environment is this social environment, the environment we set up between us. And it ends up being that every time you do research on humans, it immediately becomes obvious that this social environment is a huge part of how humans develop and how they, for example, access healthcare and what they do and all these other things. So if you want to understand human health, you have to understand the social environment. That's interesting. It gets you into psychology and psychiatry and those medical domains. And it also gets you into neurobiology. Why and how do humans think the way they think in the setting up of this? And then the other interesting thing, as I mentioned, is that humans themselves are an environment. We're an ecosystem, and we have a very active ecosystem of gut bacteria There is now 1 or 2 medical procedures where replacing the gut microbiome is an effective treatment for particular things. So there are 1 or 2 cases where this really works. There's an awful lot of snake oil and pseudoscience also in this field. And so kind of impacting this between, understanding what is going on, understanding our gut microbiome, understanding what is science versus pseudoscience is tremendously important. And that is closely related to this other transversal theme, infectious biology. Edith always stresses, we were going to do this before COVID. Unsurprisingly, when COVID happened, so much of our effort went around responding to the virus, and we did a lot of data sharing enablement for COVID, which I'll touch on, I hope, later on. But as well as the microbiome and the way they interact in the gut, there's also this business of really understanding how parasites, viruses, and bacteria contribute to diseases. And this is the work by Maria Bernabei here in the second thing of a tissue-engineered vasculature. So she can make artificial blood vessels in the lab. And she can make artificial blood vessels that look like tissue and artificial blood vessels that look like brain. And therefore, she can study how parasites interact with these blood vessels, and she can do genetic manipulation of the cells before she makes those blood vessels. And so there's a completely new toolkit coming up, which is the ability to create sort of tissue-level systems that replicate parts of the human system for infection biology. We love microbes so much that we have a particular focus on them. And so this is the business to look at how bacteria interact with their environment in all sorts of different ways. And one important thing to realize is there's an awful lot of microbes that really don't like oxygen. And so when you do something in the lab, I mean, people forget we live with oxygen all around us, and it's totally dependent for life. There's a chunk of really important life that finds oxygen highly, highly poisonous, and they just cannot be exposed to it whilst they're functioning. And so that is why you need quite a lot of focus on being able to, if you want to study those microbial ecosystems, which might be in the soil, or they might be in our guts, or they might be in cows' guts, you have to deal with this anaerobic, very strictly anaerobic situation to be able to study this. And that requires quite a lot of different mindset and technology around it. And then there are 2 more interdisciplinary areas here. So one is bringing in more mathematical thinking. Now, EMBL's always had quite a lot of maths and computer science in it, but this is formalizing that process even more and having a sabbatical program for mathematicians. I have no idea whether there are mathematicians. Are there any mathematicians in the room? Perfect. If you're a biological mathematician, this is for you. We hope to run a sabbatical program where we can bring in mathematicians for a period of time to be alongside and see the problems that biologists have. And there's a really interesting interplay here between AI and mathematical theory, where AI, which is linked both to this theory world and to the next world, data science, ends up being a great way to understand molecular biology, but often you need to have the right mathematical framework to present the problem to the AI machine. And so the mathematics of this is quite important. How do I represent an organism's 3-dimensional layout in a sensible way without going bonkers, basically? You need a good maths framework for that. And that links to this other side, data sciences. Again, EMBL has been doing this a very long time, in particular EMBL EBI, which is really the anchor institute, not just for Europe, but often for the world, for these data resources. So the reference human genome, the reference mouse genome, these protein structures, and all these other things. But we want to extend that across EMBL and make it part of EMBL in all the different ways, and we want to share the expertise about how to do this with all our member states and our partner institutes. Now, I know this is a big part of what you do here in Elixir Norway and other things. So definitely, this is all part of that same business of bringing more computational biology, quantitative biology, and data science into biology in a productive way. And one of the huge success stories has been this collaboration with DeepMind on AlphaFold. We started working with DeepMind about 5 years ago when this was an idea, and when they were looking around for interesting problems to solve. I must stress that the actual algorithm itself is really John Jumper and Demis Hassabis in DeepMind. So they're the people who came up with this. But they themselves will acknowledge absolutely two things. The first thing is that this problem was unsolvable unless there was the deep and diverse protein structure database present in the PDB, so the protein data bank. So if we had not been collecting protein structures from the 1970s onwards, they would not have been able to solve this problem. And the second rather wonderful thing is that they collaborated with us to provide everything out there for free, which was rather generous of them, and it's actually non-trivial to do. But let me just tell you about this problem. Many of you will know that the protein sequence very often determines the unique three-dimensional structure of a protein. So if you just have the protein sequence, you stretch it out, you put it in the right solvent, you give it the right ions, very often the protein sequence will refold to being a particular protein structure. Now, that's not true for all protein sequences, and it's not true for 100% of a protein sequence. There's some floppy bits and some disordered bits, but there are bits in the middle which we call globular, which do this. So DeepMind solved this problem. Now, they kind of, at one level, cheated in the sense that they used evolution. So they don't take just one protein sequence. Usually it takes at least 10 or 20, but these days it's very, very easy to get an evolutionary spread of 10 examples, just 10 different mammals, 10 different vertebrates. It is not a hard thing to do. And it's not really cheating in the sense that everybody had decided that this was an okay thing to do for a very, very long time in this game. And then they developed an AI program. It is the most complicated AI program that DeepMind, who arguably founded AI, have ever developed. And it's really interesting about the details about this. They had to reimagine the way they did certain things to solve the problem. So there's actually quite a long and complicated story about how they developed the AI system. But eventually the system produced confident predictions. Now, why are people so confident in this? The reason why is that John Malt in 2000 set up a competition for this problem. So every 2 years, this CASP competition happens, where John goes around and he asks his colleagues in the structural biology community, which proteins are you solving the structure of now? Oh, sorry. Apologies. And I'm out of time. I'm going to have to speed up, aren't I? Anyway, they are confident because of that competition, and they can solve these structures. I think I'm almost done. And we are doing AI in different areas. So one is very much image analysis. One is genomics. This is my lovely Danes, and doing AI on the Danish healthcare system to crunch the numbers there about risk prediction for this. I talked about those things. I just want to finish up touching on a couple of things. So one is training. We've always done this business of doing pre-doc courses, but we've also done, so that's about PhD students. Please recommend to your undergraduates the EMBL PhD students from a very broad range. You can be a physicist, a mathematician, probably not a historian, a molecular biologist, wherever. If they're interested, And if they're interested and they're enthusiastic and they want to learn, they really, really have a place to learn at EMBL. But we've supplemented this, and in particular, there's a new program for technology developers and engineers. So this is very much not the PhD track. This is an engineering track, and was stimulated somewhat by the scientific engineering in physics. And this is to allow people to be trained sort of on the job in software engineering or technical engineering to deliver these scientific services. And then touching on innovation and translation, we do spin-off companies. So, for example, one of them is this one from EMBL Rome. We actually have a great line in X-ray image detectors through a spin-off company in Grenoble. But we also do very deep public-private partnerships, and one around databases has been Open Targets. This has almost raised 100 million pounds, 100 million euros, probably has raised 100 million euros over its lifetime. And these are pharmaceutical companies putting their hand in their pocket and bringing money to the table to accelerate parts of the research that they want to see happen in the data sciences. And if you do drug discovery, this is an incredibly good informatics platform for doing this. And then we've got these technology development projects. For example, some of the newer microscopes from Zeiss and Laser come from collaborations or spin-outs licensing from EMBL. And then we do this integration of the life sciences. We work with lots and lots of ESFRIs, but I'd particularly highlight the Elixir aspect, which Linné also highlighted on bioinformatics. The hub of Elixir is hosted by EMBL in the same buildings as EMBL EBI, and is developed there. And I can see that I have really badly overstepped. So this is my last and final slide. Please, please, please engage with our program. Please recommend the PhD program to students or colleagues. If you're a postdoc, please, and you're interested, you're a PhD student, you're ending, or you're a postdoc, thinking about your postdoc, please come and apply for being a postdoc. You can design your own postdoc via the iPod program, which is a bit daunting, but we can help how you design that. And then in particular, use our services, so come and exploit us in Heidelberg. So thank you very much, and apologies for overrunning.
Yeah, yeah. Thank you, Ivan. I will now like to invite Trygve, the director, to join for a panel discussion. And 3 Norwegian alumni, Rein Aasland, Professor, Department of Biosciences, Faculty of Mathematics and Natural Sciences, University of Oslo. Rein also serves as a delegate for Norway to the Elixir board. And Rein worked as a postdoc at EMBL Heidelberg 1992 to 1995 in the gene expression program. And welcome also to Karina Wibe. She works as a postdoctor in Barcelona at Center for Genomic Regulation and EBL Barcelona. Karina worked as a PhD at EMBL Heidelberg 2015 to 21. And then welcome also to Øyvind Ødegaard Fogner. He works as a postdoctor in the laboratories of Harald Stenmark and K. Oliver Schink at the Radium Hospital. And Øyvind, he did his PhD at EMBL Heidelberg 2015 to 2020. You work on advanced imaging, I believe. Yeah. Good. So my first question to the panel, and maybe I can ask you, Rein, do Norwegian fellows and researchers make good enough use of EMBL?
No, not at all. Why are we doing this anyway? Of course, there's a reason. No, it's always been too low. And I suppose there may even have been times there were no one at one of the EMBL stations. But no, we don't. And we Did you say why?
Yeah, what is it due to? Is it
Well, if there was
knowledge?
if there was only one reason,
Yeah.
and it was well known, then of course we wouldn't have had this problem, because there is no quick fix, because I think there are several reasons. One is I think maybe we can discuss that later today. There's generally low mobility among the young people in Norway. We see this recruiting to the universities. Even Oslo and Trondheim University tend to recruit mostly from their local regions. In the beginning, the EMBL did different things than was common in Norway, but I think that's changing. It has changed already, so that is not a particular reason. A third thing that I think is important, there are several, is that seniors, as you said, and mentors tend to influence where the young ones go. And that's a positive feedback loop. So there were lots of people going to the Americas, US, and many went after in several rounds. So I think that's part of it as well. And of course, there's not enough information and knowledge about EMBL in Norway.
No. So too bad. So Karina and Øyvind, you were just there. Could you share with us some your experiences from working there?
Yeah, it was really intense, I have to say, and tough, but at the same time extremely inspiring. I think for me the major thing was being immersed in this culture of basic biology, and also as Ewan mentioned, the interdisciplinarity is really, I'm a developmental biologist, but I was involved with doing computational biology, bioinformatics, even theory at the end of my PhD. So I feel like my horizons really expanded. And there's just like an enthusiasm there, which is really, I don't know, it's an incredible, it's a privilege to work there, and you really notice when you leave that this, you don't have this everywhere. So I think it's a really special environment, and, all you have to do is, it's not that you have to be good enough to go there, it's that you have to be enthusiastic enough, because that's what really they value there, that you have the willingness to learn and to explore, and this is really fun.
Yeah, and also you have a very big network there. I know that we have internet and you can share data very efficiently, but if you are stuck in a problem or a challenge, then most likely you have the expert sitting in a few offices next to you. And to get this network and to learn how to work from the people that develop the new theories, it's also something that's extremely powerful when you are actually at EMBL. And something that's difficult to get if you are just accessing the databases and using the facilities, because you get the personal connection to people as well. And as Karina said, it's very intense and you are really challenging yourself, but you come out of it at a very different state that compared to when you came in.
And Rein, you were there in the 90s, so I believe you also have built a network from that time.
That's been tremendously important. By the way, could you mute the slide, please? It's extremely strong. And so my career after EMBL has been enormously influenced by the network I was able to build there. So that you said, if you go back 10 years or 20 years later, you won't see anyone. So I experienced precisely that. In the end, it was only the cleaning lady in the section I used to work in who was still there, and the Percy as well, who has been dean of PhD and so on. But then later on, this has spurred participation in new projects, and I think even it influenced why I serve on the Elixir board and stuff like that. It's been fun, interesting, and very valuable.
But as we know, the research and development going on at EMBL, it's really a common good for European research and innovation. So should we then say, is it enough that Norway and Norwegian researchers, that we use EMBL and other European infrastructures? Is that, should we be happy with that, content with that? What do you think? What do you think, Trygve?
Well, I think that, now we have seen the diversity of EMBL, and we have learned about this new program. And it was in particular when we saw this new program, Molecules to Ecosystems, we were directly asked for recommendation, more or less, by the Research Council, is this of interest from Digital Life Norway side. And it truly was. It was very well aligned with what we are doing here. And yeah, the infrastructures and availability, as you mentioned here, is of course one big thing of this. But I think that there are programs, postdoc programs, PhD programs. There is much more in it than I was aware of. So there is more than the infrastructure.
Could we also, to you also, could we also think about other types of interactions that could be developed? Are they there, you know, between infrastructures, short-term visits, strategic collaborations? Could you say more about?
I think all of those things are valid and worth doing, and we are very flexible at EMBL about how we set these things up. What I feel happens is somehow people don't realize. So just looking at some of these things, there we go. Some of these things, people don't realize the possibilities that they have. One is their career possibilities, and they think somehow it's going to be too hard to get in, yeah, or the competitions, you know, somehow you will have to studied in Germany to be a successful postdoc. It's just absolutely not the case at all. So we need to diffuse those perceptions about applying. But the second thing is coming and going. Sorry, my voice loud. The second thing is around the experimental services. I'm really, really keen for the cellular biologists to really understand the opportunity that they have. Do you want to fix something?
Yeah. So while he's getting batteried up, there are training courses, advanced courses, fantastic. Not all of them in these 6 places, but other places as well, and conferences. And those are accessible and very popular and extremely good. There we go. For instance, yeah. But and I do actually wonder whether we shouldn't have quite an explicit, like almost, you know, the Society for Cellular Biology in Norway. I don't know where that is. Or something like that. We have a biochemical society. Biochemical, yeah. Yes. You know, the most number of people who are obsessed with cells in a room, and find an opportunity because I think a lot of people just sort of, you know, they probably assume that they should go to Stockholm or Oslo for this thing, and they don't realize they can just put their hand up and also go to Heidelberg. That would be my instinct. I might be wrong.
Also about putting your hand up, something that's, I think, even more under-communicated than the PhD and the postdoc positions is that it's also possible to do a master or even a bachelor project in the EMBL labs, for instance, in my lab, in the Jan Ellenberg lab. They will never announce these positions, but if you are motivated and you take contact with the lab that you're interested in, EMBL has always, it's so flexible that it can facilitate these things. And I never heard rumors of a master student applying in our lab, even.
But it's information. You need to know and learn early.
Yeah, and these positions will not be announced. You have to raise your hand up, like with many things for EMBL.
Yeah. So when we were master students, this is a long time ago, we organized our own excursion, and we chose Germany, and we went for a week through Germany, and one of the places we stopped was Heidelberg. This is 2 years before Norway became member, and I still remember Kai Simmons, one of the leaders there, said, go home to Norway and say that Norway must join, and I think some of us were influenced by that, and actually had that in mind when we chose where to go later on. Knock on the door.
Yeah, take initiative. That's the home message. But this month, Norway's new long-term plan for research and higher education were published. And some of the priorities in that plan was health, life sciences, enabling technologies, and including fundamental research. And the plan also mentioned that fundamental research within marine, agriculture, and food sectors need to be strengthened. And also Norway supports the EU's 5 missions, as it were. So, so to you, would Norwegian life sciences benefit from collaborating with EMBL and the new program in areas where we are strong, as it were, within marine sciences, within health? Would we, thinking of the plan, the priorities that Norway has now, would it strengthen us to work more closely and with the ambitious new program?
Yes.
Yeah.
The answer, I think, is yes, of course, it is. And it is also this international perspective on this. And I think you see now with in this plan that you mentioned, it's if you go to the last part of it, it's very updated. It seems like it was written 2 weeks ago, because it also takes the crisis that we are in right now into consideration. So more sharing when it comes to infrastructure will be more and more important. Biotechnology is more and more relying on heavy infrastructure, which means that the costs are not only for personal costs, but the running costs are increasing incredibly. This is more and more discussed. So sharing of infrastructures, not only nationally but also internationally, I think will be very important. And this has to be coordinated.
Yeah. And then, Trygve, what role can Digital Life Norway play towards EMBL, would you say?
Well, I mean, we are a national center, and we represent many scientists. Some of them are here, and even more are out there. And we also represent more or less the most important institutions and universities and institutions in Norway. You mentioned Earth Biogenome Project as an example. There is a Norwegian node that was recently started. And this initiative was taken more or less from DLN, and we were asked by the Research Council, can you take a coordinating role in this? Because if we are going to support this, it has to be a national initiative. It has to be collecting all interested, relevant players, so we make it into a truly national initiative. And this had to happen very fast, and this was done. It was a signed letter of agreement among all those institutions. So this is one example where DLN really can play a role. And I was thinking that it could also be similar here. We discussed this also back in the board, that, yeah, maybe we could take a role because we have common mutual interests, as you mentioned, with respect to training of scientists, transdisciplinarity, and of course the life science biotechnology in common. So for sure we could play a role in this.
Yeah. So I don't know about the time now. We should probably round up, but maybe you would like to...
Maybe could just a brief comment to
that. Yeah. Please.
For high energy physics at CERN in Switzerland, there is now a Norwegian, I guess it's a virtual center, and there's a clear address and a home base for Norwegian participation in the CERN laboratory. And I think one of the handicaps we have, there is no similar body or organization or anything in Norway for the EMBL system, so maybe that's precisely what could be done here.
And if you want to, we should, I didn't say this, but Øyvind and Ryan and myself and Katrina are doing a workshop tomorrow morning. So if you want to hear more about this, you get to see us again tomorrow if you want to. Come, ask anything.
Yeah.
And prepare for that. Have questions and bring them.
Yeah. So good, Ewan, you stole my last point there.
Oh, sorry.
So
really
for those who are more interested, yes, please come to the workshop tomorrow. I don't know whether you would like to say something about it, but...
I mean, you can also approach us in person at any time if it's easier just to speak one-on-one. If you feel like it's intimidating to apply to a place like EMBL, then I was terrified, so please talk to me. Also outside the workshop. So.
Good. But I think we end this part on EMBL then. So thank you to the panel.
thank you so much to Ewan and all of the panelists who were able to share some of their experiences from EMBL. And of course, we will have 2 workshops tomorrow, both with Lisa from Danish Board of Technology and EMBL. So start thinking about which workshop you'd like to attend. It's time for a little refreshment break, so please join us out in the hall, and we'll be back here in about 25 minutes. Alrighty, we are back. I hope you got a bit of refreshment, a bit of a break, a bit of a bite, perhaps. So we are really privileged to have two speakers from the Bio and Emerging Technologies Institute, based in Addis Ababa, Ethiopia, at Digital Life 2022. The two speakers have worked on a wide range of biotechnology challenges with a deep motivation to deliver value for society, to understand societal needs, and to co-develop solutions. In line with this year's theme, their work has used a transdisciplinary approach, using two-way dialogue with stakeholders nationally across Ethiopia, as well as with innovation partners elsewhere in Africa, North America, Asia, and Europe, including right here at NTNU. First, I'd like to introduce Yordanos Ali, who currently works as Associate Researcher 2 at the Bio and Emerging Technology Institute, known as BETIN. She's the team leader for enzyme technology, and her research interests include production of enzymes for industrial applications, mainly for industrial waste products to develop feeds and to reduce costs for society. And she also leads a number of hands-on trainings. After her presentation, we will hear from Brooke Esaya, who's researcher at BETIN as well, who works on a number of interdisciplinary projects at the intersection of biotech, nanotech, and computation. He has researched the development of silicate-based platform chemicals for industrial applications and worked in close collaboration with the private sector for biomedical applications. And both of the two speakers are founding members of their institute, which started back in 2016. So, with that, I'd like to introduce Yordi. Please give her a warm hand.
Thank you. Hello, everyone. I'm so happy to be here with you guys to share what we have and get some points, what you are doing so far. My presentation is going to be on what we are doing on Ethiopia regarding on biotechnology. When I heard the previous researches, most of the institutes are the big ones, and they're spending almost more than 50 years on this biotech, and that are our grandfathers, or more than that, maybe. So our institute is, that might be the younger one. The overall coverage of our discussion right now is going to be what's BERIN, what is working on, and how the society responds for the work that we are working regarding on this biotech. The picture you're seeing is the established members for BETIN. Initially, its name was EBTI, but now it changes to BETIN. So it is established in 2016. It's almost only 6 years, that means. So what I'm going to present for you is that we're trying to work within these 6 years. It's just to know, is there anyone before in Ethiopia or having idea of biotech research is doing so far in Ethiopia? Anyone who have been collaborating or anything? You, okay. So you have some ideas on what's working in Ethiopia. So mostly when we say there is biotech research in Ethiopia, people say, you starting it? That's why I'm asking you guys if you have any experience on doing researches in collaboration with any universities in Ethiopia. So it's have a mandate on doing researches, high impact researches, because small researches or initial simple researches are doing in universities, higher institutes related to this biotech, but the mandate for BETIN is on high impact researches and having a product and giving service for society by using the product that we produce from our research. The other is giving trainings, focused technical and short-term trainings. We give hands-on trainings for undergraduate student, MSc student on the molecular techniques or any biological techniques on research. And now, last year, we get a mandate to give advisory role to give advice or consult different governmental and private sectors. So these are the mandates that the institute is established. Before going to our research cycle, we have two directorates. The first is bio, the other is emerging technologies. In the bio, we have this medical biotechnology, agricultural biotechnology. The agriculture is into streams, plant and animal biotechnology. The other is health biotechnology and bioinformatics and genomics. And in the emerging, we have nano biotechnology, reverse engineering, and material science directorates are working on the emerging side. So we work a lot of researches in convergence with physical science and the biological science. So how are we going to select our researchers if anyone comes to our institutes, or how are we going to deal with or select our researchers? The first is on the site survey. Our researchers or the institute researchers go to different sites of industries or agriculture zone to see the problems there. So within that survey, we bring our question of the research, and through review, or some industries also come to our institutes to do on some problems. And the other is a national request. As I told you, it is from governmental institutes, so the government itself gives some problems or research topics to be covered. Then the defense will go on, as common in any research areas. The research work is, we work it in our lab or in collaboration with other institutes, as you know, to work on this biotechnology researches. You need budgets, and everything cannot be covered within a single institute. So what we do is that we collaborate with other institutes, international or national institutes. So some part of the work will be done in our institute. The other will be done in the other institutes. We don't stop because we don't have something. If we get something, we work it. If someone or other institutes have it, we'll go there and do what we can do. Then having a result, so it's related to science society first, because what matters first is to discuss on the results, and is that really now ready to deliver for end users or to disseminate to the end users. That's how our cycles works. Afterward, what I want to show you is that just simply exemplary researches that we are doing so far is not the only, but it's just to highlight what kind of researches that we are focusing on. Maybe ours will not be that much complicated or on the molecular level, but it's just to show you we are in the beginning of trying to work on researches that can contribute something for society from the biological basis. So this one is a research working on looking for yeast-tolerant, ethanol-tolerant yeast strains. Why we're doing this is that while we visit the sugar industries, there are different sugar industries in Ethiopia. So they, after production of this sugar, they got a lot of byproducts, molasses, having 40% of sugar. So that sugar will remain as a waste. So they try to convert it to energy form, and they produce ethanol in their industry. They have ethanol plants. So the strain that they were having and use it for ethanol production was imported from India industries. So they use it for around 10 years. After that, it was not effective, but they were initially use it. So we try to look for indigenous yeast strains that can help them to increase their production. These are just the picture in the culture that we have the yeast strain. So as you think, the one in the blue is that the one they have, the E strain is the imported one. So it works only on 12% of Brix concentration, that means sugar concentration in the molasses. So our strains are still working on 50% and 20% Brix. That was more than their expectation. So we are in the pilot work one now, and after maybe 6 months, they will be applied to their industry. Sorry. The other model research is also based on the isolation of these yeast strains for local. This is a traditional drink, mostly common in Ethiopia. It is made up of honey, pure honey, and after the fermentation, it's going to be changed to alcoholic, and it is very common. But it was not a standardized one, or the production step and fermentation step is different from area to area. So we want that to commercialize it and export it. So it needs to be optimized, and the standard have to be set for this production. So what we're doing is that searching for yeast strain, specifically from the honey. We find those strains from the original honey. So now it becomes standardized and commercialized in the market. As you already heard, I'm a team leader for enzyme technology team. Regarding these enzymes, we are on the startup. What I mean is that we are not producing this enzyme in the large scale, but we're trying to produce them in the lab. We work on protease, amylase, cellulase initially, but now we start working on phytase and xylanase because there is a big problem in the feed industries. They are importing a lot of enzymes that can be applied on the feed substrate. So we're trying to work on finding indigenous different bacteria strains that can be applicable on the enzyme production, but we are not still on the scale up. I'm sure after getting people here, we'll collaborate on production of these enzymes and scaling up. After this, some of the designs or research that I'm going to present you are not focusing only on the biological system. I know that most of us here are from biotechnology or biological science, but not all the researches has not to be based on the biological. What I mean to say is that whatever the sources, what we want is to solve what the society is in problem. So we try to convert different wastes from different industries to the energy form. So this one is the sugar industry use the bagasse or the straw that they have after production of sugar for energy source, and they burn it and change it to steam and use it for the industry energy source. So they have a bagasse ash produced in the industry, so we want to convert that bagasse ash to produce briquette as a charcoal to be energy source, and also we design the material department design instruments that can be possible or used to produce this briquette, and we train young people who have now a job to produce this briquette, to produce it and to commercialize in the market. This one is also using the bagasse ash that's produced in the sugar industries for production of these cement blocks for construction. The construction material cost is much higher in Ethiopia, so we're trying to replace the cement by this bagasse ash. So the blocks are already starting produced. This one is almost similar to the prototype that's produced from plastic wastes. So we try to convert the wastes that we have in the industries in different forms to be used as partition board. This is for partition board purpose. The other is there is a lot of wastes in the coffee industries. As you know, Ethiopia produces a lot of coffee and export it. But in the production process, there is a lot of coffee husk that is deposited in the industry. So these wastes have to be converted to fertilizer. So simply we use epigetic earthworms species to convert this coffee husk to the fertilizer. Then we train young people in the area to produce this fertilizer and to use it for their lands. That's how we're doing. This is a clay. I think you have experience of eating maybe injera, Ethiopian traditional food, that we are eating it 3 times a day, maybe. If we're staying here more than a week, maybe I am not the one who's presenting here. Because we can't live more days without having it. So, but the production process or the material they're using for preparing this injera takes a lot of energy to be hot and to process. So the material department doing is that changing the composition of the clay so they can produce, make injera with little energy. This is just to show you that mostly researchers are coming together and saying on the molecular level, on micro level, on enzyme. But for a country like us, it doesn't give a sense whether you say DNA, RNA, what the shit. So what are we going to do with it? You see, they question what their problem is that. So one side we do at the molecular level, the other side we are always looking for what we can do for society directly that they can feel. The other thing that I want to share with you all is that we're trying to develop national bioeconomy strategy. We want to change the country economy to the bioeconomy. So we were part of this Eastern Africa Bioeconomy Strategy Development Team. I was a member of that national working team. But when it turns to East Africa, Ethiopia will be out of it. So what we're trying to do is now developing our bioeconomy strategy, and because we think that we got a lot of things to work on this bioeconomy. Ethiopia is very rich in the biodiversity. There's no question, because the ecology zones, agroecology zones are very different, and within that agroecology, we got a lot of biodiversity that we can be used as a source for bioeconomy. The government commitment is very high on working on the bioeconomy. We want to change the system to biological system. R&D training institutes are a lot in the country. Around 38 universities working on this, giving a course biotechnology and related courses. So the trained manpowers are also available, even if it's not that much skillful. And indigenous knowledge, we're thinking that every science comes from the indigenous knowledge. So we want to dig out what the indigenous peoples have in their hands, so to exploit what they have and change it to a product. So we have all these sources, use manpower as much. When we're sizing this, we're not saying that everything is in a straight line. We have a gaps on funding, on awareness. What we mean by awareness is when you say Ethiopia biotechnology, everyone's saying, are you saying GMO? We're not interested, but biotech is not all about GMO. It's not allowed in Ethiopia. There is a bit gap using these GMOs, but the wild ones still have application in the biotech. And the biotech means a lot different from a GMO. So we're trying to create awareness regarding on what we mean by economy, why we need it, what we have, and what is the advantage of using that. We're trying to use social medias, institutional medias, and the big medias also. We used to convince people what we mean by that. And also when we go to farmers, we try to communicate them in the way they can understand. They never know what bioeconomy is, but we just to discuss with them what they have and how they convert it and how it's going to be economic source for them. And capacity for lab equipment and reagents, hard currency, of course there is a problem. But as I told you earlier, we try to work in collaboration with different national and international institutes. We're good on that. We think that we believe we are good on collaborations. I'm sure I'm going to have a lot of collaborators from here and to work a lot on this bioeconomy and biotechnology. So we collaborate with a lot of institutes, and we're so lucky on that, having here a lot of people from different institutes having similar interests with us. So our all interest is doing researches from scratch, or the complicated one, the easiest one. What we try to work is something what we have. and looking for something better. That's what our interest research area. And having a product, so changing society well-being. Unless we reach to the society, and for our institute especially, whenever you present something as a proposal, then what you have for the society. If it is for publication, you can keep it whenever you are in the universities. But when you come to national research institutes, you have to have something to be given for the society, so the society have to trust us that we are there to do something for them. That's what I want to say, and thank you for your time in that session. Thank you very much.
Okay, good afternoon. I'm very happy to present what Jordi started to as a continuation. I know you do very advanced researches, and you are interested to hear our stories on undertaking research on biotech research with multiple challenges in a developing country. And I'm also very happy to hear the story of EMBL, because I, with a background in physics and nanomaterials, 3 years ago, when I want to make a research on biotech, I was recommended to read 2 books. One of them is about written by The Double Helix by James Watson, who invented the DNA, and the other one is the 8 Days of Creation, about the birth of molecular biology, how European laboratories were in a fierce competition, reinforcing each other in the creation of this molecular biology. In one of the stories, how the labs, the biology labs in Scandinavian makes the nitty-gritty of this sample preparation, and on the other hand, on the British and French laboratories, they do this XRD analysis, and especially taking the blood from horses, and so that this myoglobin structural analysis goes through to finally to Nobel Prize. My story, Jordi presented the general part. I zoom into two research activities which we did in collaboration with international partners, and how these things bear fruits. The first one is collaboration to develop a robust molecular diagnostic platform suitable for low resource countries, where I develop isothermal heat baths for lamp using beeswax nanocomposite as a thermal buffer, in collaboration with Professor Johannes Kabisch, formerly in Germany, now working in NTNU, and the other one is Dr. Jenny Molly from University of Cambridge. WHO recommends diagnostic devices or systems suitable for low-resource countries should meet the ASSURED criteria. ASSURED is an acronym for affordable, sensitive, specific, user-friendly, rapid, and robust equipment-free, and deliverable to end users. In this regard, when we say affordable, simplified in instrumentation, and sensitive, it has to be a molecular diagnostic system with it can amplify to 10 to the power of 9 copies in 30 minutes. This is a brief summary of what the device or the platform we envisaged, and what we tried to show in some forms, and primer designs and multiplexing capacity. And the other one, it helps us to no complicated detection mechanism, but with visually detected using pH-sensitive dyes, and the time to result is some 30 minutes. Not well equipped with facility, and it doesn't take a grid electricity, because it's very hard to find in developing country. And the other one meant to be point of deliverable to end users when, say, point of care service. So diagnosis comprise normally two, usually the hardware and the reagent component. I will come to the hardware part in the next slides. But for the reagents, where we worked together with our international collaborators, The reagents include enzymes, buffering agents, oligonucleotides, and primers. The key reagents for molecular diagnosis are enzymes, and these enzymes usually requires up to minus 20 degrees centigrade to put it, and it's very hardly to get an infrastructure which can maintain minus 20 degrees centigrade, let alone this infrastructure. It is very hardly possible to get electricity in most part of our country. So to overcome these things, two approaches were proposed, and one is the sport display technology, and the other with Professor Johannes and that of University from Texas in lyophilized long BST for polymerase, and then we plan to make assay optimization protocol for COVID. Yeah, for COVID gave us a very hard time for the global community, but it is a blessing in disguise for us because we were planning to make the hardware for LAMP for long, but when the COVID comes, this idea of making the LAMP hardware get some funding, and with that we realized the device. So the general consideration for heater design was optimal assay temperature. There is some optimal temperature at which the assay is optimized to amplify, and it has the assay tolerance to temperature fluctuation has also very high implication on the specificity or on the sensitivity, because for viral samples, plus or minus 0.5 degrees centigrade fluctuation from that constant temperature of amplification may result in 50% decrease of sensitivity, and it also requires minimum holdover time, maximum warm-up time, and sample volume and sample formats are very important parameters. So talking of the Ethiopian case, for example, talking of the temperature variation, Ethiopia's altitude vary from minus 140 below sea level to 4,900 above sea level, meter above sea level. So the average ambient temperature correspondingly varies from 2 degrees centigrade and up to 43 degrees centigrade. So in this whole range of temperature, to maintain a constant temperature, it's a very challenging task. And had it been in a developed country where there is electricity available, we can use a Pelcher effect material as in PCR, but that one is equipment intensive. So that is ruled out from applying the Ashur criteria. We opted for a phase change material. As you know, when materials change their phase, they maintain some constant temperature, which is a latent heat of fusion. So for that, we selected beeswax. Coincidentally, this beeswax, the beeswax melts at the temperature where most of the amplification temperature for bacterial and viral sample. So with effective thermal insulation, we hope that we can realize it. This one. And the other requirement beyond this phase transition point, it has to have high latent heat of fusion, and it should have high thermal conductivity. Unfortunately, the beeswax has low thermal conductivity, but I will talk how we get over this challenge. And the remaining things, it fulfills very well, those mentioned here. So as you can see from this diagram, this is a differential scanning calorimeter. This deep area is where, with a constant temperature, it absorbs the differential. With a constant temperature, it absorbs this amount of minus 3.32 milliwatt for just 1 milligram of beeswax. That means when we calculate the latent heat of fusion is about 231 kilojoule per kg. Just to change in 1 degree centigrade, it has to absorb at least this amount of energy. That means 56 times more than that of water-filled heat bath. So water is. So we want to take advantage of this huge latent heat of fusion, and to increase its thermal conductivity, we make a copper oxide nanoparticle using sol gel preparation, which is a very facile method, and we confirmed that it is through X-ray diffraction, especially this 35, the 31 and 38 peaks are unindicative of the copper oxide nanoparticle. So we blended it with beeswax so that we can we engineered a composite phase change material, which is nearly, as you can see from this figure, it's nearly in liquid and semi-liquid state, indicating of the engineered phase change material has a sharp melting point at 63.2 degrees centigrade, and which is a desirable characteristic from the point of assay amplification, and provides one more degrees of freedom to optimize efficient heat transfer mechanism via the interplay of convective and conductive heat transfer. So, and it also may be applicable, because I saw some paper as well that from paraffin wax they try to make lab-on-chip diagnostic format. You can also, because it has very high latent heat of fusion. Also, the other challenge is to make it high throughput, to use the standard molecular biology microarray, which with a 96, with a 98, with 96 tubes. So for that, we have to assure that it has the thermal homogeneity has to be maintained. So for that, we have to include some fins. As you can see, the heat is supplied just below. As you can see here, this is the heat supplying things, and the wax is deposited here, and so this one can assure that with the fractal geometry can assure that the uniformity of heat transfer. So on top of this, we put our assays to test it. This is maintain an isothermal temperature, that's constant temperature for 18 minutes, even after the insulated chamber lid were opened, and we tested for some for COVID. And now, when I say platform, what I mean by platform is once we establish the hardware and the enzyme stuff, so it's a matter of changing from malaria to plant diseases to other animal diseases and to all other diseases. It's about changing the primer. So once this, by platform is just like something high ground, so that you can see the problems in every direction, and once that problem is coming, so we can design the primer, so we can optimize to this temperature up to 63 degrees centigrade, so we can implement it. We are not short of problems when it comes in developing country when it comes to diseases. So this service. The other one I go through quickly, curbing the adverse public health and socio-economic impacts of mycotoxins, which is being undertaken in collaboration with Beijing Genomic Institute, even the African Union Center for Science and Industrial Research in South Africa, and two from Ethiopia, and at one part on the instrumentation aspect, we also get a collaboration from North Carolina Agricultural and Technology University, and University of Dar es Salaam from Tanzania is also involved. What I learned yesterday that mycotoxin is not as such a problem in Europe for two things. First, most of the products, agricultural products which came from Africa, have a stringent regulation, because about 0.5 microgram per kg of things is discarded, not to be exported from Africa. But when it comes to Ethiopia, we don't have this facility. We only use for export commodities only this characterized, or this measurement is used. But Africans have also the right to have a safe food, and to get this safe food, they cannot afford, because one measurement or one quantification takes about 3,000 birr in Ethiopian context, and there are only two 2 labs that provide this service for the population of 120 million people. So, yeah, mycotoxin is highly toxic metabolite produced by molds in contaminated foods, represents serious health risk for human and animal health. And mycotoxin contamination affects up to 85% of staple foods in Africa, because the Africa climate is a bit favorable for fungal growth. It's humid and warm. When fungus get humid and warm environment, it's spread very fast, and so because of that, it affects the African economy very badly, because even in Ethiopia, 30% of the coffee fail to be exported because of the presence of fusarium, one kind of aflatoxin, and difficulty to decontaminate due to its temperature resistance. And if animals feed a contaminated feed, and it goes through all the way, and you can get in the milks of in the milk taken from that cattle, you will see still the aflatoxin. So it doesn't degrade through the digestive system when it goes through. So with that, child takes this milk, and prolonged exposure to even minimal amount of the mycotoxin is resulting in lately reported finding is that it leads to child stunting. So in a way, aflatoxin is killing Africa's future in this sense. So there is a Pan-African initiative to deal with this problem, and my institute, the Bio and Emerging Technology Institute, is an executing agency for PACA, Pan-African Partnership for Aflatoxin Control in Africa. So we take part on that initiative, representing Ethiopia. So we designed around, as you can see from here, around 10 projects, but these are the ones who get through funding. The first one is biocontrol of aflatoxin using ataxogenic Aspergillus flavus strain. It is in collaboration with Beijing Genomic Institute and that of the Pan-African PACA. The idea of this research is to get ataxogenic Aspergillus flavus, so that we can crowd out those toxin-producing strains at the end of the harvesting season. And the other one is using lactic acid bacteria to inhibit fungal growth. And the second approach is a chemical method, combating aflatoxin with neutral electrolyzed water, which is a very simplest method, and it's affordable. As I will show you in the next slide, this method make use of only salt and water, and so that farmers can use post-harvest in the post-harvest even at their tablewares. And the other one, notable one, is this one: adsorption of aflatoxin from contaminated milks using bentonite and zeolites, led by Dr. Wandimagay Mamo. It's also there is also a collaboration with a university from Portugal on this regard. As I told you, milks, minimal exposure through milks led to child stunting. And the other one is detection. Also is one way of combating its impact. The first one on this regard is detection of aflatoxin B in food sample using smartphone-based nanosensors. This not pushing that much, but this one we get through the. It is truly interdisciplinary research, portable machine learning based mid-infrared spectroscopic sensor development for screening mycotoxin contamination in field. And we have collected enough data from the biotech aspect, that is the metabolomics, how the host and the fungal interacts, and the intermediate molecules leading up to the things, so that we can pick some fingerprint molecules to teach or to make learn the machine, so that the sensor is a very simplified one. And this is somehow at some stage, and through funding, if we get funding, we are about to realize the device instrumentation aspect. The new synthesis follows, as I said earlier, it's very simple. There is a membrane, ion exchange membrane. So we start with a 0.2 molar of sodium chloride, and we apply the direct DC voltage of 12 volt and a current of 1.1 ampere. And we did the electrolysis for 20 minutes, and our solution was pH is 7.1, redox potential 974 millivolt, and with this, the most impact come from the synergistic effect of the pH, the redox potential, and the available chlorine content. And once we prepared this one, we tested with a duplicate sample for 10 to the power of 0 for the stock solution, and then for 10 to the power with the serial dilution 10 to the power of -2, and in all of the cases 100% inhibition of the growth of the fungus, and this is corresponding control samples, and as you can see, it grew. We didn't stop here. We just tried to make a prototype by 3D printing it. As you can see it, we 3D printer, we built this one. It can produce 1 liter. So 1 liter, we mean at a time from what we produce, we can make 100 liter of it, because as you see from the previous slide here, 10 to the power of -2 is also effective on, this is some contamination. I think this one is a real one. This is we took the picture very late time. After 3-4 days, there was nothing of this sort in the sample. So with 1 liter, we can produce about 90 to 100 liters, so that the farmers put it in their post-harvest silos, so that they can control or fight the aflatoxin exposure. And this one that I told you, thank you so much. If you do have questions, thank you.
Thank you so much to Brooke and Jordi for your presentations. We are now going to take another short break. So please feel free to go outside, and we'll be back at 4:15. Alrighty. I think I always say alrighty. I don't know if that's. Anyway. So now we've had another break, and it's time to introduce a Digital Life Norway projects, one of the Digital Life Norway projects that's also based here at NTNU. I'd like to introduce you to Prostomics. And I will start off this session by asking for a show of hands. How many people in the room know someone who has had prostate cancer? Wow, that's quite a few people there. Then I think you might be quite interested to hear about the Prostomics project and reflect on the question, what should cancer scientists be doing to build a dialogue with the community that their research addresses? So in a few minutes, we are going to welcome to a chat here on stage to discuss this collaboration with May-Britt Tessem, who's Associate Professor and Research Scientist at NTNU and the PI of the Prostomics project, which is funded by the European Research Council. And she will be joined by Ulla Kinsat, who is a sociologist and retired researcher, as well as a former prostate cancer patient who serves as a patient research partner in Prostomics. The chat will be moderated by a friend of Digital Life, Giovanni De Grandis, who's a philosopher here at NTNU and also the coordinator of the Affino network, which focuses on new ways of generating and sharing knowledge and foresight through transdisciplinary collaboration. But before we have them on stage, I would like to present the video of Prostomics that Digital Life created, coordinated by Christopher, our communications advisor. So take a look at Prostomics.
My name is May-Britt Tessem. I'm a researcher and an associate professor at the Department of Circulation and Medical Imaging at NTNU. And my research is focused around prostate cancer and molecular multi-omics technology on clinical tissue samples. The Prostomics project is an ERC starting grant. The goal of this project is to identify biomarkers and understand molecular mechanisms on how prostate cancer becomes aggressive and life-threatening. So Prostomics is based on analysis of larger patient cohorts of high-quality frozen prostate tissue that is donated from surgery of patients with consent. And we drill frozen and targeted tissue samples from several places within the prostate to study heterogeneity within the tumor, around the tumor, and also in normal appearing cells. So from each selected sample, we cut very thin sections of the core, which is used for the various omics analysis and histopathology. So we have recently focused on omics method that provides spatial information within a tissue. We want to understand the molecular mechanisms within the tissue microenvironment. By doing spatial analysis, you can go deeper in your analysis and you can identify biomarkers that haven't been identified before. So an important method we use in the Prostomics project is MALDI-TOF mass spectrometry imaging, where we receive molecular maps of metabolites and lipids and peptides almost down to cell level. So this method uses laser ionization directly on the tissue, which is coated with a specific matrix, which is sprayed in layers directly on a tissue section. The specific machine can be extremely fast because the laser shoots rapidly over the tissue surface, and one small tissue sample can be finished within minutes. We also use spatial transcriptomics, where we can see gene expression of thousands of genes like a map on our tissue samples. We also use different microscopy techniques like confocal microscopy, where we stain different compartments or specific proteins of each cell in the prostate tissue to get really high detailed 3D images of the selected proteins. So by integrating the data from the multi-omics results, we aim to understand more on the molecular mechanisms that separates aggressive from slow-growing types of prostate cancer. It's especially exciting because I designed the project myself. We have built up this activity for years here in Trondheim, And we also have excellent collaboration with the St. Olav's Hospital and the Biobank One, where we have all the high quality tissue. I hope that the patient first will benefit from this, and also good biomarkers will benefit the clinicians that have better tools to give the right treatment at the right time. And I also hope that the society in the whole can benefit from this by reducing overtreatment and reduce the costs and the resources needed for treating patients.
So, please join us on stage.
You and Ola. Where will you sit? Shall I take this seat and you and Britt sit over there? The place you in the middle.
I'm in the middle. Okay.
Yeah.
Actually, I was invited for a sofa interview, but.
Yes, we were promised a sofa. But thank you very much for the invitation, and I hope that this debate, this discussion will be interesting, and it has been very interesting for me to try to do this and to meet Ola and Britt beforehand, and I think they are very, very interesting people with very interesting things to say. And I would like to start this chat by asking you if you can describe how the involvement of a patient representative in the project came about.
Yes, actually I was planning not to have a user participation in the prostomics because it was the ERC project, basic research, and I thought, oh, that's okay. But I was approached by the Cancer Society, who asked me if I wanted to have a user representative on project level in a basic research project as a pilot project. And I said, not really. I think I was afraid of the workload, and actually in the beginning I said no. But they were really eager to make me have a user representative. And before they asked me, I have met Ola, so I kind of knew him. And then they said, Ola will be your user representative. And then I said, okay, maybe I'll do it then. So we had discussions back and forth that they said that this will be no extra workload because he will just be participated in your group meetings, and there will be nothing that you have to do to have user participation. So this was kind of the beginning. Yeah.
And since you have mentioned the fact that when you discovered that the proposed patient representative was Ola, that made change your mind. So can you tell us how did you met Ola before? How come that you two were already?
Maybe I should.
Maybe Ola can take that.
Because
Yeah.
after 5.5 years ago, I read an article in the local newspaper about this big project, an interview with Mai. And I am a member of the board of the local prostate society. So, and we should have a meeting at Brekstad in Fosen outside Trondheim, open meeting. And I proposed that we should ask Mai to tell about the project at that meeting. So for some reason, she was driving with me because it was a Thursday and I should go further on the weekend out there. We were well known. So I think that was the start, that you know me. And,
we got to know each other
Yes.
on that trip, so I knew more what I was expecting.
And Olaf, from the perspective of prostate cancer patients, what kind of expectations and hopes do you have from projects, basic science projects like proteomics? Obviously, it's not something that will produce an immediate cure. So, from the point of view of patients, what is really important and interesting about that?
You know, it's several reasons. First, it's the reason that research is necessary to get the new methods, new medicine, and new kind of treatments. But back that, you need basic research, which is quite different from applied research. Also, the role for user. But we found out, I suppose that many of you didn't quite understand what Mai was talking about when she was in the video. I have been there for 5 years, and I still have large problems with understanding what's going on. But that's because my role is not to be a researcher. It's to be being the making Mai and Maria, who is sitting down there, you are, mind them of that. What people are they researching for? Who is going to have the results? That's me, and other old male with have had prostate cancer.
So one main point is to remind the researchers that they're not just working on tissues, but there are human beings
yes,
to which these tissues belong.
Yes, and they should not be afraid of prostate cancer as females. There's no problem.
And Britt, you said that at first you weren't enthusiastic. So can you, as frankly as possible, tell us about your feelings, your thoughts, your worries about having a patient involvement in the proteomics project? So, and also, once you changed your mind, what were also the hopes about it? So, what were the fears and what were the hopes?
Yeah, my fears was the workload, because I was thinking that in a project like this, it's hard to do user involvement as I have read about it in the Cancer Society, because I was seeing myself just trying to present difficult things to Ola and make him understand difficult things, and that there will be no dialogue. That was kind of what I was fearing, and that I needed to arrange things extra with my time. So that was actually my biggest fear. And my hopes, you said, I didn't really have much expectations, but we have already started user participation in the bigger group, where we also do MR imaging, but that's more like patient-near research. So I thought that that would probably be easier for the user to understand. So my biggest fear was that it will be just a one-way communication from me every time we meet, because then it will be, the workload will be on me.
And as a matter of fact, I did not have any clear thoughts about what was I going into when we started 5 years ago. but we found relatively quick out that to participate at the regular project meeting once a month, that was the way to do it.
And the Cancer Society at that time promised me that you can do whatever you do at your meetings. I'm not changing the agenda or anything when he's arriving. And we're still doing that.
Yeah.
And sometimes I think you can be quite frustrated when you're into the details. But the only thing I do is maybe update you on what happened from last time, because you are not there every week, right? You are there once a month. That's also quite often. But I think the only thing I do is just updating in between what's going on. Now we're discussing the details of blah, blah, blah. And that's also good for the group, I think, that we do that.
I think so, too. And I report a bit back to the board of the Cancer Society, telling what's going on.
Prostate.
Yeah. And I think that we are involved, and it's known by our members. Mai and one of her colleagues, Tone, was last year in a member meeting and told about the project. And Mai has also been in the Nordic or Scandinavian Congress, which was held in Trondheim some years ago, 3 years ago, I think. Yeah, where we both was.
We were invited.
We were invited, yes. So it's how to spread what's going on and what we are working for.
So that's one of the positive sides then. That.
Yeah. And Hola, how were you feeling when you started your involvement in this project? You told us that even now you are not completely understanding all the aspects, so I guess that you must have felt at least a little nervous, or were you having some worries about being able to act as an intermediary between the Prostate Cancer Society and the project?
Yeah. I didn't know what I was going into. I have no idea. But I thought, I'm curious. I'm a retired researcher in quite another field. But I still am curious of what's going on. And I want to learn. And I get a lot of opportunities to learn. Even I don't understand everything. And so, but I think we have found a nice way to do it, to work together. And I am very aware that I am not a researcher in this project. I am a user participant. Yeah.
And Britt, can you explain why the Cancer Society was so important a player? Because you had already your project funded by the ERC, and then we may wonder why the Cancer Society comes and asks you something, and why this comes as a sort of request that is hard to resist. So what is the role of the Cancer Society from the perspective of a researcher?
Yeah, for a cancer researcher, the Cancer Society is very important because they fund research projects, of course. And they are, they have a requirement that you have to argue on your user participation when you apply for money. And I think it was half a year or maybe a year after, I was actually funded a Cancer Society project. And at that time, I was actually very happy that I could say that I had user participation, maybe on a higher level than anybody else at that time. And it also was a big discussion, I remember in 2017, which project should have user participation, should people working in a dish have user participation, and all this discussion around that everybody was kind of requiring that the Cancer Society should kind of define what they wanted. So I think that's also maybe why the Cancer Society asked me to do kind of a pilot project. But we haven't really discussed it with the Cancer Society afterwards. So we don't have any measure on it except our experience.
Maybe I get an opportunity on Tuesday next week,
Yeah.
because then it's a day conference in the middle of Norway for user representatives in research. And it's the Cancer Society in the middle of Norway who are standing behind the research. So maybe I get the opportunity to ask about it.
You should, yeah.
And Ola, as compared to the Cancer Society, which is a big society that funds research, can you explain instead what is the role and the kind of interest in research that the Prostate Cancer Organization has? And so what are their expectations from engaging in research, and why they think it is important?
Yes. I think for all the projects and all the research, we are interested in these topics because we want that fewer men get prostate cancer and that fewer men die from prostate cancer. And the methods, I remember it's 15 years ago I was at surgery. They are not doing it the same way today. We are getting new techniques. And this is an all-time running process. We need research to improve the methods, to improve the medicine, and to get. And we are living longer today than we did a generation ago. So it works.
Yeah.
But we have to be patient sometimes.
And in practice, how was the involvement of Ola implemented in the project? So when and how you interact, and how has the process evolved, if it has, during these years?
Actually, it started with the arrangement with the Cancer Society that he will come, and we kind of decided that you can come every fourth week, every once a month. And in the beginning, we actually discussed it a lot after the meetings. Is this useful? Did you get anything out of it?
Yeah.
I was a bit nervous, I had to say. But you said, yeah, I think it's interesting. So, okay. And then you really want to still continue to come? And then say, yes.
Yeah.
Okay. And then we...
Yes, but you also took me around to the lab.
Yeah.
So I saw the drilling that we saw in the video. So I've been around, and I think I've been well known with all the people, the researchers that works in the project. And yes, so I think, and it's very nice to come, because I'm taking very well, what it could take godt imot.
Yeah.
Yeah.
You're very welcome.
Yeah.
Yeah.
So how long did it take before both of you were thinking that this was working and this was worth doing?
Maybe half a year.
Yeah.
Something like that.
And also because I started to think about what are the positive effects of having Ola here, and we talked about it.
Yeah, we did.
And I don't know, I was thinking about it, and it gives me kind of a motivation that Ola, as a former patient, he's really interested in the things I'm doing, even if he sometimes thinks it's difficult.
Yes.
And I think also it's kind of motivation for the researchers, the master students. They see that Ola is coming, and he's a former patient, and this means something that you're doing. It's not just your master project or just your. I think this motivation is good.
Yes, I think so.
And it's a reminder for us, as you say.
Yes.
Diving up from all the details that we're working with. And also, the communication part,
Mhm.
because Ura have invited me several times. I mean, it was COVID, there
Yeah.
was a bit silence then, but he's invited me to talk to like the general public, and that's also important in our ERC project to communicate. And that's something I report back that I do, and also to understand the patient perspective. That is something I wouldn't have if I was just only working in the bench. Yeah. So I think it's important, and it makes me reflect about it more, I think, because he's there.
And I think we are very agree upon the goals of the project to improve things. What we not so much agree about is the fear of over treatment, which you might mentioned, because the prostate cancer as a community in Europe, also the urologist, what is pronounced in English, now want screening, like it's for breast cancer. But then, of course, there's a problem that people can have prostate cancer which don't need to be treated. You can live with it. So and therefore, it's very important with the project that are getting better methods to divide between aggressive prostate cancer that should be treated and not aggressive prostate cancer, then you can just follow up over years. So, but we live with that. We are a bit disagreeing on this topic. No problem.
It's politics. Yeah.
So from the point of view of the project, I understand that the main benefits have been the motivational one of seeing the real relevance of your work for patients, for real human beings, and the networking elements, that this has opened the project to the possibility of communicating more also outside academia with patient community, and this is a valuable thing. But from the point of view of patients, instead, apart from keeping up with what is happening in research, is there any other benefit or any other thing that the prostate cancer organization has appreciated about your reporting, your keeping them updated?
Yeah. I'm not quite sure what you mean, but the society is concerned about what's going on. We have used representatives in a lot of projects, but this is quite a different project. This is a long time basic research project. Mostly it's applied research, but we try to participate with user representatives in as many projects as we can. So
there is one positive effect that I believe, but I don't know because we don't have research on that. And I asked Ura because I think the openness showing what basic researchers do, I think is important, and especially in these times when I think basic research is kind of pulled down, and I think maybe that Ura represent and just show and talk about what we are doing in a basic research project, all the things we are struggling with. He knows about GDPR, he knows about ethics committees and all these things that we that we struggle with. And maybe also the skepticism against research. Maybe this openness can give something out to the general public that
yes,
kind of show how we're doing it, how we are using the money that is raised in the Cancer Society by by the people.
Yeah. So would you agree that participating in the project has helped you to see the relevance and the importance also of basic science, that it has made it less remote from...
Yes, maybe, but that, today is most belief. We don't have facts. So we should have research on it. Does user participants in project improve the project? It's a nice idea to search for money to do.
Actually, I was asking somebody, I don't remember who, about this when I started having Ola, that maybe there should be some research going on when I'm doing this pilot. But they said, oh, yeah, that would be very interesting, but we lack funding, so we cannot do it. Yeah, so that was the answer, yeah. So I also think to measure the real value, you need to do research on that.
Because I think it's about 5 years that the Research Council, the Cancer Society, Helse Midt-Norge, the regional I've been occupied with that. It should be user representatives in the research. And now it's time to start an evaluation. Is it really doing something? I think maybe there are differences between the basic research, which is a long time going process, and applied research that maybe have a time perspective on 1 year or something, maybe 2 years. So, because in applied research, you can say if you can't write this introduction to the patient that you ask to participate. You, as a patient, we know better what kind of formulations that motivate patient or an earlier patient to say, yes, you can get data from me.
Yeah,
that's
actually
quite different.
Something you have helped us with,
yeah.
Like patient consent and how we write that in a good way. Yeah, so that's something you have helped us a lot with.
But since you have mentioned this possibility of measuring the usefulness of this involvement, well, my very strong impression from this conversation, but also from talking with you before, is that both of you think that it is valuable, that it has brought something that is worth the effort. But do you think these benefits are easy to capture, to pin down in something that is objective or measurable or a sort of clear indicator, or do you feel that they are a little more intangible and maybe it's better not do it too much research because
I think maybe that's more
in
your league of research.
To show why.
No, but as I said before, we need research. We need new knowledge to get on the every long process of being better. And of course, it's hard to say from one project because basic projects will sometimes work and find out this is not relevant. That's also a part of the research process. But then we know that. So we can go on with alternative methods. So I think it's valuable to, and it's valuable to get involved from the user perspective. And that it's also, I was reading a bit on the Cancer Society's website earlier today, just to see what are they meaning. And they are pointing to that we can have perspectives as users that the researchers hadn't thought about, giving new problems to research on. I don't think it's that much in mice proteomics, but in applied research, I think it's more common.
And have there been any major challenges or things that proved frustrating or difficult in your collaboration?
No, I didn't have any frustrations, I have to say, but except from the beginning when I was accepting a high workload. But I mean, just having you there, there's no work at all.
It's just an occasion that we were driving together before we started this, and we would get known to each other. Had a nice lunch in Brekstad before my wife came with the Hurtigbåten to the windows. And Maya and the rest of the board went back to Trondheim, and I went to Stocksund. So I think this was not planned as an introduction to being a participant in user representative in the project. So but so is life.
But I think also that you or I is probably a special user representative, and you have research background, and you kind of understand the research probably much better than the general population of users. So maybe that. And also that we had good chemistry was a good thing.
Yeah.
So no solutions and conclusions on.
It's very good that there is this good chemistry, because you told me that you have some different views on some political topics around cancer research and cancer prevention strategies. So can you, but nonetheless you respect each other, and this is very good, because it's I think it's very important that this kind of disagreement are understood from each part, so that we can see that there are issues about which it's reasonable to have different views, and it's sort of a reasonable disagreement. And can you tell me how you see the politics of cancer research and cancer prevention, and if this engagement with each other has changed a little your perception of the politics of cancer research and cancer prevention?
I guess what we mostly don't agree on is, of course, the screening situation on prostate cancer, the PSA screening. You have heard about that. Because I don't think, my personal view is that we don't have tools enough in the clinic to actually separate those that are aggressive from those that are indolent and grow slowly. And that's why if we have screening, I think we will do treatment on a lot of more patients, and also those that maybe didn't need it. But that's something that we cannot measure either now, because we have also wild screening. But it's coming, I know that, because the European Association of Urology, they decided that it's a recommendation now. And then, so it's the politicians who have to figure out if we're going to have the screening or not in Norway. And since Sweden already started, I think also Norway will come. And this is also something that the Prostate Cancer Association are really activating and really want this into the society. And that's our maybe political disagreement, but
But that's what take the word further on. And if we get screening, then many more men will be diagnosed with prostate cancer. And then it's even more important that projects like Prostomics are succeeding in finding tools to split up the population of prostate cancer men in aggressive cancer and not aggressive cancer. So, because I think too that is coming, and that means it's even more important when it's coming that you got good results.
Yeah, I think it's coming pretty fast. So we'll see if I can do something really quick.
And that will be very good because it's a well-known dilemma in public health that when you have preventive screening, that can issue into action. The risk is that this action will have a lot of benefit for some people who will be saved by this intervention, but will impose a cost on some patients who would have not needed the intervention. So there is always a trade-off between imposing a burden on some people to create a great benefit for other people. But if your project works and achieves these results, maybe we can remove this trade-off and only treat those who need treatment and not impose any cost to other people.
If it hadn't been that difficult, so I mean, we have been trying, yeah, but we'll see.
Okay.
Yeah,
I think we are probably approaching the end. So, do you have maybe a very quick suggestion for making involvement successful to any project who is starting? Any recommendation?
For
how to
patient or
run,
former patient getting into it?
Yeah.
Be curious. Ask questions. And yes, I think that's, and if you know something about research, that's not a disadvantage. But you don't need to have it. We have many colleagues that use representatives that are not researchers or have been researchers. But when you are retired, you have more time. And I think it's very interesting to use that part of my time on being involved in research as you as a representative. If I can say, for many of you, you never get prostate cancer. And I have noticed when I'm on stand that very many men are just, it's a bit taboo to speak about prostate. But the wives and the daughters, they are more interested in that their father or husbands should test themselves to early detection. I had some handouts like that I leave here from the Prostate Cancer Society in English that describe what prostate cancer is. So I just leave this at 10 copies that I had home. So, but it's been very interesting to be here.
And I also want to say that I really appreciate you, Ola, that you are curious and that you want to spend your retirement time, precious time, to be within this project. So when this project finish, we'll find another project. Yeah.
But you're a very good host.
Thank you.
Yeah.
Thank you very much, Ola and Britt. It has been a very interesting conversation, and it has been a real pleasure to meet such remarkable people as you two. Thank you very much.
Thank you.
Thank you. Could we have all of the speakers from today come back onto the stage? Just quickly. We have something for you. And while they're coming back up, I just want to share my hope that you learned something new and maybe that you got some inspiration from different perspectives on digital biotechnology today. And I think all of those who were presenting on your different areas of expertise and different projects. Yes, and I look to my colleagues for a bit of support. So on behalf of Digital Life, we want to present you with a small token to say thanks for the time you've taken and that you remember Digital Life, yes, afterwards. Let's give them a round of applause. Thank you. So as I mentioned earlier today, and as you know, we have 2 workshops tomorrow. I'm going to give you a bit of information about that now. We have the EMBL workshop that will be taking place in this room. So if you're interested in EMBL, you can come back here immediately at 9. We don't have an opening session, so you go immediately to your workshop. EMBL will be here, and the workshop with Lisa on public engagement will be in the room called Brassesparken, which I understand is some kind of soccer move. And that's upstairs in the hotel. So you can go to Brassesparken at 9 for the public engagement and here at 9 for EMBL. You may have also noticed that we have posters this year. This is backed by by popular demand for young researchers to present their research in poster format. So please make sure you take a look. We have a dedicated poster session tomorrow from 11 to 12. So the people who brought the posters will be standing next to their posters, available to ask questions. And tomorrow, starting at 11 until 12:30, we will also vote on favorite posters, and we will have a prize for the voter's choice of 5,000 kroner for your project. So make sure to take a look at all the posters tomorrow and send in your vote, which we will give you more information about between 11 and latest at 12:30. So yes, information about the workshops you can find on the conference webpage, so you can still make up your mind which one you'd like to go to. And dinner is at 6 in San Siro number 3. So see you there, and thanks for a nice day 1.